A 14-year-old in Guadalajara followed an AI-generated lab protocol step by step, sourced from a tool trained on publicly available genomics databases. The protocol worked. The organism it produced wasn't dangerous. This time, the AI had no idea why. This isn't a story about a teenager. This is a story about what happens when a tool that sounds authoritative is trained on data sets that have never been audited, and about the gap between the AI gave me instructions and those instructions were safe. That gap is where public health policy currently lives. The genomics data those tools are trained on, GenBank, UniProt, Open PubMed repositories, community contributed sequences on platforms like NCBI Sequence Read Archive, contains retracted studies, unvalidated experimental results, and synthetic sequences uploaded without provenance verification. The AI doesn't know which is which. Neither do most of the people using it. In 2023, researchers at the University of Toronto's Acceleration Consortium tested 4 commercially available AI-assisted chemistry platforms, including tools built on large language model architectures similar to GPT-4, and found that when prompted with ambiguous synthesis queries, all 4 generated procedural outputs drawing on retracted or questionable literature without flagging it as such. That finding was published in Nature Machine Intelligence. It wasn't covered as a biosecurity story. It was covered as a hallucination story, which missed the point entirely. The problem wasn't confabulation. The problem was confident retrieval of real contaminated data. In February 2024, a biohacking community forum on Discord with roughly 11,000 active members began circulating a protocol for producing a modified Bacillus subtilis variant using consumer-grade CRISPR kits from companies like Addgene and The ODIN. The protocol was generated by an AI assistant, specifically a wrapper built on OpenAI's API, marketed as a DIY synthetic biology coach. Members tested it. Several reported results consistent with the predicted phenotype. None of them flagged at the original training reference. A 2019 paper from a Chinese research group had been retracted by the journal ACS Synthetic Biology in 2021 due to unreproducible methodology and concerns about undisclosed conflicts of interest. The AI had no retraction database integrated into its retrieval layer. It retrieved the paper as a valid source. It presented the protocol with full procedural confidence. No one in the forum checked the primary source. They had no reason to. The tool sounded like it knew. This is the failure mode that biosecurity frameworks are not designed for. Not a deliberate act, not a sophisticated actor, but distributed confident error at community scale. Stop for 1 second. Every major policy conversation about AI and biosecurity, the White House Executive Order on AI Safety from October 2023, the Bletchley Park AI Safety Summit, the biosecurity provisions in the EU AI Act focuses on one threat model, a bad actor using AI to deliberately design a novel pathogen. That threat is real, but it's also legible. Governments know how to surveil for it. Export controls on dual-use equipment, monitoring of gene synthesis orders through the International Gene Synthesis Consortium. These are imperfect, but they exist. The threat model nobody has a framework for is this: thousands of people acting in good faith, following AI-generated biological protocols trained on unvetted data, producing unpredictable outcomes at a scale that overwhelms any surveillance system designed for the deliberate actor. Scale changes the math completely. One person making an error in a DIY lab is an incident. 10,000 people making the same AI-recommended error in parallel across 18 countries is an epidemiological event. And nobody's existing reporting system is connected to Discord. The core bioinformatics databases that train most AI biology tools are not curated for AI training. GenBank, operated by NCBI at the National Institutes of Health, contains over 3.9 trillion nucleotide bases as of 2024. It accepts submissions with minimal experimental validation. The assumption built into the architecture is that scientific community norms, peer review, institutional oversight, replication, would filter bad data before it reached the archive. That assumption was built for a world where accessing GenBank required graduate level training and institutional affiliation. Now, a wrapper around GPT-4 with a GenBank API key costs $12 a month and requires no credentials. The database didn't change. The user population did. Most AI biology tools don't generate information from scratch. They use Retrieval-Augmented Generation, RAG architecture, which means they pull from indexed scientific literature and database records, then synthesize a response. This is actually safer than pure generation, but it introduces a specific failure. The AI's confidence is coupled to its retrieval quality, not to the underlying truth of the document retrieved. A paper retracted for data fabrication, if it remains indexed, gets retrieved with the same confidence weight as a robustly replicated finding. The retraction notice, often published as a separate document in a different journal section, frequently isn't indexed at the same retrieval priority. Researchers at MIT's Computer Science and Artificial Intelligence Laboratory published an analysis in 2024 showing that across 5 major biomedical RAG systems, retraction integration was absent or inconsistent in all 5. That's not a flaw in a single product. It's a structural gap in how the field is building these tools. Remember the Guadalajara teenager from the opening? His tool wasn't regulated because it wasn't classified as medical software. It wasn't classified as a laboratory instrument. It was classified as an educational AI assistant, a category that under both FDA frameworks and EU AI Act risk tiers carries the lightest compliance burden. The gap between what a tool does and what category it's assigned to is where oversight disappears. The FDA's Digital Health Center of Excellence has jurisdiction over AI tools that make clinical decisions. The CDC's biosurveillance infrastructure monitors reportable disease events. But an AI tool that helps someone design a home experiment that produces an unreported biological outcome that falls between every existing regulatory perimeter. This is not a gap anyone designed. It's an emergent gap created by a category system built before the tools existed. There's a specific kind of danger that human systems are very bad at responding to. We are excellent at mobilizing against visible threats. An outbreak with a named pathogen, a cyber attack with an identifiable source, a bad actor with a stated motive. These threats have edges. We can draw a perimeter around them. The harder threat is distributed error. Diffuse, well intentioned, technically enabled, and structurally invisible until it reaches a threshold that overwhelms our response capacity. The people in that Discord forum weren't trying to cause harm. The developers of those AI tools weren't trying to enable harm. The researchers who published the original retracted study probably believed their results when they submitted them. Every single node in that chain operated with reasonable intentions and incomplete information. What we've built inadvertently is a system that amplifies incomplete information at a speed and scale that makes correction difficult before consequence. And the deepest discomfort here isn't about the technology. It's about what this reveals about how public health infrastructure was architected. It was built to respond, not to preempt distributed, low signal, high volume error. That's not a technology problem. That's a systems design problem. And technology is running ahead of it faster than any regulatory body is currently tracking. So what actually happened with the oversight failure? The experiment went viral because the AI-generated protocol worked for a benign application with a benign organism. That's the version we know about. The thing we cannot know, because no system currently exists to track it, is how many AI-generated biological protocols produced anomalous results that nobody reported because no reporting pathway exists for a home experiment that didn't quite work the way the tool predicted. The surface question was, how did an unvetted protocol propagate at scale? The answer is through a confidence interface layered on top of an unaudited data set delivered to a user population that had no reason to distrust it and distributed through platforms that no existing regulatory framework touches. A harder question, and this one doesn't resolve cleanly, is this: if we built real-time retraction integration, mandatory provenance tagging, and risk-tiered regulatory classification for AI biology tools tomorrow, would that be sufficient? Or have we already reached the point where the distribution infrastructure, Discord, GitHub, open API wrappers, moves faster than any compliance framework can track? Because the answer to that question determines whether what comes next is a policy solution or a fundamentally different kind of problem.